welcome back to the cell signaling course. We were discussing in previous class about the GPCR, G proteins, type of receptor, type of signaling which goes downstream of the receptor activation. Today in the class we will see how different routes are taken downstream of the GPCR. Also we will be discussing how G alphas of different types are working to ensure that the diversity exists in this signaling. Let's start with the G alpha signaling. We have been seeing the G alpha responses downstream of G alpha S, G alpha I and G alpha Q. We have seen those responses in the previous class. Now we are discussing the G alpha 12 and 13, which is the fourth class of G alpha which is present in the system. Bulk understanding is that there are four main types of G alphas. We will see how there are variants of that exist in today's class. So as we have been noticing that signaling starts downstream of the GPCR by binding of the ligand here, bringing the G alpha beta gamma heterotrimeric complex in the vicinity of the receptor, activation of the G alpha by binding or exchange of GDP with GTP, and then activating a next molecule in the line. Here in case of G alpha 12 and 13, the next molecule which is activated is ROGEF. And if you recollect, ROGEF will ensure that Rho GTPases are now in on situation, which is basically mean they are loaded with GTP. And this beta gamma subunit can independently induce the signal. So downstream of G alpha 12 13, there are activation of ROGEF, which ensures that Rho A GTPase is active. Then this Rho A GTPase works through ROCK or Rho kinase, and which basically regulates the actin dynamics. Also, multiple of the metabolic functions is where this G alpha 12 13 signaling is involved. Just for information, that G alpha 12 13 is not that name which is interchangeably being used. G alpha 12 is one type of G alpha and G alpha 13 is one type of G alpha. They are clubbed together because mostly they are working downstream of the with the GTPase of Rho family and involving the actin cytoskeleton as well as metabolic functions. So we will see some examples of G alpha 12 13 signaling. It has a big role to play in the metabolism, where it will detect small molecules like ATP or its versions, in the sense that ADP, AMP, further the base adenosine. So if you look at a scenario where this G alpha 12 13 is working, and we are taking example of a particular process where this ATP and adenosine are serving as a ligand. So if this is my receptor which binds to ATP, it will work through the G alpha 12 13 and it activates the Rho A GTPase. What this Rho A GTPase will do, it will ensure that relevant signaling continues and set of right transcription factor are activated to transcribe set of genes. Basically in this case where we are discussing the ATP as a ligand and this pathway is activated, consider the heart muscle cells where this thing is getting activated, where you will see molecules which are like TGF beta are activated. They are made to mediate a fibrogenic response. Whereas when this GPCR, where you have G alpha 12 13, where adenosine is serving as the ligand, it again activates the Rho A GTPase and the signal is again going inside the nucleus, activating transcription of molecules which will be involved in fatty acid biosynthesis. So basically this G alpha 12-13 seems to be working with multiple different types of molecule. It could be small molecule like this. It also will be sensing the lipid like molecules, amines and peptides and so on. However, the final response is towards the actin cytoskeleton and gene transcription. We will see more detail of this in coming slides. So this was an example of G alpha 12-13 participating in the metabolism. The other example which is there available for G alpha 12-13 is in the fat metabolism, and how the condition of the organism allows switching between one type of metabolism process versus the other type of metabolism process, and how different G alphas are involved. So again, consider a scenario here where you have this GPCR present on the membrane, and this is another GPCR which is present on the membrane. Now the two conditions which are being sensed is that whether it is a fat rich diet which has to be metabolized, or the individual is undergoing exercise and there is need of energy. So what is understood is that when you have fat rich diet, generally response moves to this one, which is G alpha 13 mediated, and when you have exercise, the response switches to this G alpha 12 mediated. As we have understood that first set of molecules which will be activated are Rho A GTPases downstream of these things. Then here some of the hypoxia factors are activated, because when you are under exercise you may face hypoxia like situation. Eventually what it will translate into is the fatty acid oxidation for energy providing. And here in this case what happens, that you will see certain molecules which are getting activated, one of the molecule called NFAT, which ensures that oxidative fibers level is reduced, which mediates more like a storage of the fat rich diet or fat in the system. Here fatty acid oxidation goes up, thus it will provide more energy. So in some sense what will happen, that energy response is more towards G12 and less towards G13, and fatty acid storage response is more towards G13 and less towards the G12. So what you notice is that it could actually play a very important role in ensuring that system is responding as per the metabolic condition of the organism. What has also been understood lately about the G alpha 12-13 is their role in solid tumor, where it basically engages in multiple or touches base with many more signaling events which are happening over there. So this receptor which will bind to G alpha 12-13 and activates the Rho A, which will work through the rock, and eventually it will cause the proliferation, migration, the hallmark of tumors and so on. Then this is the main role which it is playing. It also works through the enzyme which is called GSK3 beta, glycogen synthase kinase. This inhibits the glycogen synthase kinase activity, which impinges on to the transcription and helps in multiple processes. Eventually, all these things which are happening through different route, like it also responds to the JAK-STAT pathway. Somewhere it merges on the JAK activation, and thus again it kind of generates the response to the cytokines and so on, which will basically ensure that there are more migration, more proliferation and so on. Basically tumor will grow more in size and more solid tumor will be generated. So combining multiple of these responses, it will ensure that solid tumor formation is kind of promoted. Also in that case, it will work through the cadherin molecules which are present on the surface and involve in their signaling, which is mediated through the GSK3 beta kind of receptor, GSK3 beta kinase and so on. So basically G alpha 12-13, apart from the canonical RhoA activation in the process of where there is metabolism concerned, fat metabolism concerned, or in solid tumor growth and proliferation is concerned, it is also affecting different other pathways which probably may be running in parallel inside the cell. Now let's discuss some of these things which is about the G alpha families and the GPCR family, its abundance and so on. What we have understood until now about the G alpha families, and which I highlighted in couple of previous slides, that there are 4 main types of G alpha. One of the first of those classes is called G alpha S. This G alpha S, I am trying to draw a rough phylogenetic tree. So if all G alpha S, which is clubbed together in one part of this tree, you automatically understand that opposite side will be class 2, where you have G alpha I, because this was stimulating cyclic AMP and this was decreasing cyclic AMP. So if you make a phylogenetic tree dendrogram, you will realize that these 2 types of G alpha find very distinct position in this tree and which are diagrammatically opposite. Then you have another big class of G alpha, the biggest class of G alpha, which is G alpha Q here, where you will see that they are ensuring that multiple of these G alpha Qs are present and grouped together. Here you will find that G alpha Q is responding through PLC, DAG, IP3 and calcium. So this is one cluster which you can see. And then you have additionally the cluster which is here, which is G alpha 12-13, and we have just seen how they are important for metabolism and ensuring that the RhoA kinase activity is activated and actin cytoskeleton is regulated accordingly. Its dynamics is regulated. Recently there has been studies which identified another class of G alpha which is poorly understood, but that has been considered as G alpha 5. More and more studies are being done to understand what exactly, where is this molecule involved. Then there are plant GTPs, plant G alphas. They have very unique property. They are in nascent stage of understanding. There are few molecules which are responding through GPCRs, because if you recollect, most of the responses which are present in plants is mediated by the receptor-like kinases, which were under the class of receptor serine threonine kinases. Here basically you have few G alpha, you have few GPCRs and so on. That study is still continuing. But what we understand in case of animals, that maximum response, because this is the most abundant type of gene family where 800 and more genes are coding for GPCRs, and there are many more which are coding for the G alphas, associated molecules and so on. And they have been divided into 4 major class, with 5th class also coming up, which we are waiting to gain more understanding. Now if we look at the structural analysis, how it has proceeded over time. So if you recollect, I told that Robert Lefkowitz and Brian Kobilka, they were given Nobel Prize in Physiology or Medicine in 2012 for their extensive study on GPCRs, and they were largely working with the beta adrenergic receptor. So if you just look at the type of receptors which are present, so out of 800 or more genes which are coding for GPCRs, almost 80% belongs to class A, which is rhodopsin type. Which is involved in the light perception. So almost 80% of the genes are involved in or almost 80% of the genes which are coding for GPCRs, they belong to class A, and thus are of structurally similar to rhodopsin type receptors. But not only that, there are many more genes out of these. If I represent this entire 800 genes in this circle, around 30% and more, they are orphan receptors, which means that we still do not know what is the ligand for them. And in the remaining, you have very different kind of signal molecules. It could be lipid, it could be amines, it could be peptides, proteins, small molecules, which are relevant with the sensory perception. So apparently there is a bias even in the GPCR genes, that what they are going to look like and what kind of process they will be mediating. And more so in recent time, the studies which have been performed on the structural aspects with the help of now cryo-EM kind of structural analysis and prediction of the structure using AlphaFold. More and more input is being received on structural aspect of these GPCR. If we simply look at the way structures have been solved for GPCRs over the years, in around 2000, there were almost only 4 or 5 structures which were solved. And as of today, we have almost, or as of 2020 I should say, there are almost 70 structures which are solved. Out of those 70 structures, more than 50 are actually from this class A receptor. So because of their abundance, as well as for the understanding which has developed over the years, there are more and more class A GPCRs have been understood in the system. So if we summarize how GPCR signaling looks like, the ligand binds to the receptor, it activates G alpha, depending on which G alpha the signaling moves forward. G alpha S or olfactory type activates adenylyl cyclase, makes more and more cyclic AMP, which binds to the protein kinase and activates it. This protein kinase A phosphorylate Krebs and helps in the transcription. Then other one which is opposing, having opposing function, G alpha I, it decreases the activity of adenylyl cyclase, increases the activity of phosphodiesterase. This leads to the increase in the linear GMP like molecule or nucleotide, and less of cyclic molecule, which affects the current which is flowing in the photoreceptor cells and affects the visual perception. We saw these examples in the previous classes. We also saw that how G alpha Q works through PLC beta and activates itself. Then it basically works on the PIP2, makes diacylglycerol and IP3. Together they work to activate protein kinase C. The DAG directly works to activate protein kinase C, and the calcium which is released helps in the process of activation. And this leads to the release of calcium molecule, which depending on cell type will mediate a function. But what we realized in the example we took with respect to G alpha Q, that calcium excess inside the cell was inducing exocytosis, and that exocytosis was for the neurotransmitter which was sending signal to the neurons connected to those cells. And what we have understood today, that G alpha 12-13, they work through Rho GTPases by activating the Jeff-like molecule. Once Rho GTPases are activated, they activate ROCK kinases, and these ROCK kinases work with the actin network. We have also seen how they are playing independent roles, which is not directly impinging on the ROCK, but rather going through GSK beta or working through some JAK-STAT pathway and so on, to ensure that homeostasis is maintained. And in case of G alpha 12-13, we saw it was important for how it was important for fat metabolism, responding to ATP or adenosine-like molecule, responding to exercise where energy consumption is happening, or fat-rich diet where fat has to be stored. Now, when it comes to the removal of the signaling, or rather termination of the signaling, we have two different aspects which play important role in GPCR signaling. One is called desensitization. What basically it means, that the receptor is no more sensitive towards presence of the next molecule, meaning it is not catalyzing the next molecule. What I understand or what I suggest by next molecule, in case of G alpha S, adenylyl cyclase, or in case of G alpha I, phosphodiesterase, or in case of G alpha Q, the PLC. So system is present over there, G alpha is GTP bound, but it is not recognizing and activating the next molecule. What happens over there and how the process moves forward, that the ligand bound receptor is signaling, G alpha is carrying out its signaling process, but once the signaling has reached its goal, there is a feedback inhibition which works. Whether it is about synthesizing the molecule which will inhibit this thing, this reaction, or producing something which ensures that G alpha is no more available to the next molecule. And one of the things which happens over here is the phosphorylation of this C-terminal tail of the receptor. This can be carried out by PKC, which was generated in one of the signaling, pKa, which was generated again, activated again in one of the G alpha S type signaling, or the availability of these G protein receptor kinase or G protein regulating kinase, right? They do this phosphorylation, and all these three molecules are serine threonine kinase. So basically what they will do, they will target specific serine residues or threonine residue in the cytoplasmic tail. Once this phosphorylation happens, this is a signal for these molecules which are called beta arrestin. This phosphorylation of the cytoplasmic tail of the GPCR is a signal for beta arrestin binding. Again, I am trying to bring parallel here, which I have told earlier, that phosphorylation dephosphorylation status of a protein induces or some sort of interaction, or sometimes it decreases a given interaction. So here what you notice is that phosphorylation of cytoplasmic tail is inducing the interaction with beta arrestin. Once the beta arrestin has bound, again these kinases can work and phosphorylate beta arrestin also. Now you have a complex scenario where G alpha is available in GTP bound form, But because beta-arrestin is present, it is no more available for the next molecule. If I say adenylyl cyclase, if I say PLC beta, it is not available to them. Thus, they cannot be activated. No activation. So, this step of beta-arrestin binding and making this G-alpha bound receptor insensitive to the presence of these next effector molecule is basically what is called desensitization. Now, once this desensitization has happened, things should move forward to ensure that signaling completely ceases to happen. And for that, what happens is called downregulation. Because there is a chance that this may be reversed and signaling continues. For that, what happens that this entire complex is internalized or endocytosed. So basically what happens here, that the arrestin which is present over here, that helps in the process of ubiquitination. So basically you will see that receptor is ubiquitinated in response to this beta-arrestin binding and the phosphorylation which is happening. I told you that this beta-arrestin can be phosphorylated by one of the kinases, and one of the kinases which is promptly working on the beta-arrestin is called BARC or beta-arrestin kinases. They phosphorylate and this situation is recognized and you see the ubiquitination of the receptor happening. Now this ubiquitination, what exactly it will do? It will also induce the ubiquitination of receptor as well as arrestin will happen, which will be a signal for the downregulation of the receptor. Here you would notice that ubiquitination in general, when this terminology comes into mind, we always think about polyubiquitination. And this is a degradation signal. But in this case, we are having monoubiquitination. And this monoubiquitination is a signal for endocytosis. So a part of the membrane where this entire complex was present is internalized and the components are recycled. The beta-arrestin, it may undergo degradation because it is ubiquitinated. If it is further ubiquitinated and become polyubiquitinated. If it remains monoubiquitinated, it will be recycled back, ubiquitin will be released, and you will see the receptor is getting back on the membrane. G-alpha is recycled on the membrane, not in association of the receptor. The ligand is either cleaved off or removed from the receptor. This phospho tail is also removed by phosphatase activity and beta-arrestin comes back to non-ubiquitinated state. So basically you have removed the receptor from the cell surface, and that is where it is called downregulation, in the sense that number of receptors which are present on the surface are reduced. That's the downregulation of the receptor. So in essence, what will happen? That your receptor will be phosphorylated, it will attract beta-arrestin, this beta-arrestin will be phosphorylated and ubiquitinated. This basically is a desensitization. Now this molecule which is ubiquitinated, monoubiquitinated as a receptor complex, it will be internalized and components will be recycled. Most of them, except that ligand which could probably undergo degradation inside the lysosomes and so on, or endocytic compartments. So what we have understood in today's class is that G-alpha protein of particularly class 4, it activates the Rho kinase ROCK and mediates actin cytoskeleton dynamics, as well as it is involved in the metabolism of fat and other small molecule, where we know that if this G-alpha is not working, you will end up having metabolic disorders like fatty acid metabolism, or it has been shown to be working in solid tumors as well. Further, a new family of G-alpha has been identified. More and more studies are being conducted on that, so we'll have more information of them at some later point of time. Further, the GPCR activation and its function, these information are coming more to us with more and more structural analysis being done on them, and what we have understood that a lot of structure which is coming up is coming from class A GPCR, which belong to the rhodopsin family. Further, this GPCR signaling utilizes a unique method of downregulation where beta arrestins play a very important role. They bind to the C-terminal phosphorylated tail and activate the ubiquitination process, which allows the downregulation by endocytosis. And this process of C-terminal tail getting phosphorylated is mediated by these special class of specific enzyme for the GPCR, which are called G protein regulatory kinases. Thank you.